So we can start. Thanks, Smita, for this very nice introduction, because you have explained different details concerning mainly the plants that are very important to take in mind, because now I will shift on the more chemical, purpose, but you have to keep in mind that this chemical is available in the cell, in the plant cells, and it is important to have the idea of what have been discussed before by Professor Smita. So, in this context, one of the first lecture you asked me to do concern to explore the complex world of the specialized metabolites. For myself, I don't use secondary metabolite, which is an old name of this type of metabolite. Usually now we use more specialized metabolite, and you will be conscious of that with what I want to present today. So concerning this one, my first lecture is the complex world of these specialized metabolites, and the three questions I want to address, and also to try to find answers, are why plants produce it is dangerous. Specialized metabolites, you have now a good idea of why. I will just mention some details. And the two other questions that are very important: where in the plant these metabolites are produced, where they come from. Obviously, you have seen that they come from primary metabolisms, but how are linked both of these two metabolisms, and after, at the end, what are specialized metabolites? And in this case, I will decipher terpenes, alkaloids, and phenolic compounds. So, as it was explained by Professor Smita, plants are not able to move, so they need to develop personal defense systems. And you have seen that this defense can be against biotic or abiotic stresses, such as, for example, biotic insects, microorganisms, and also herbivory that are able to produce several effects on the plants, but also the plant competition that it was explained. On the other hand, other type of stress, such as abiotic stress, have also a very high impact on the production of secondary metabolite or specialized metabolite, particularly temperature, that can able to produce different things. This type of signal can be used also in the process of plant cell cultivation, but also UV can induce several responses for the plant. They are able to produce a large amount of metabolite that are able to protect them from UV, better than us, for example. And also snow, wind, and also salinity. Salinity is one of the main abiotic stress that are able to induce several type of specialized metabolite in order to permit to the plant to resist to this different concentration of salinity in the soil, mainly in the soil. So both of these type of signals can be used for inducing this type of metabolism in the plants, and all of these are not able to induce the same type of metabolic pathway. So means that if insects are able to induce one type of metabolite, UV induce another type of metabolite production, and also salinity induce a third type of metabolite production. Means that the plant have the ability to adapt completely the synthesis or biosynthesis of metabolite to the type of the signal she receive during the life. And this is very important, because when you harvest plant in order to screen the possibility that they are able to produce this type or not of metabolite depends on the situation in which the plant is. you can completely miss a large part of the capability to produce some specific metabolite. So it means that, in fact, we need to put the plant in several types of environment in order to have a complete idea of the property of production of metabolite. And in fact, here are the list of the type of mechanisms that are able to induce this type of metabolite. But what it is also important to have in mind is that while primary metabolite, lipid, carbohydrate, and so on, are common to all plants because they are essential for the development of the plant, specialized metabolites are more specific compounds, which means that, in fact, if you find a plant that is able to produce one type of metabolite, it can be the only one that are able to produce it. You can't have the possibility to find this metabolite in other plants. Precursors, yes, but the final molecule will be more species-specific compounds. They are not essential for the growth, that's true. But they are essential for the plant to survive to any modification of the plant. This is why we have moved the name from secondary to specialized, because when I asked the question to my student, what do you understand by secondary metabolite? Always they say to me, but because they are not essential. Yes, they are, because even if they are not possible for the plant to produce them, they can't survive to any modification of their environment. So this is why now the community, use mainly specialized metabolite or natural products. This is for commercial purpose, natural products, instead of secondary. And what it is interesting is that all these molecules have biological activity for the plant, because they have been synthesized for one purpose, but also for us. Depends on what we are looking for. So it is interesting also when we screen the possibility of the plant to produce some specialized metabolite, to have also a very large set of test of biological activity in order to see in which domains this molecule could be used. Pharmacological, anti-cancer, cosmetical, so anti-aging, antioxidant, and so on and so on. There is lots of tests that need to be developed in order to estimate the biological activity of this molecule. Okay for the plant, but for us, it will be another type of activity that we are looking for example. So I think we have not discussed this type of necessity, but before starting a project for producing one metabolite, we need to characterize them for all the activity. So this is important to have in mind also. So by this way, it is important to see that sometimes, for example, if some are antibacterial activity for the plant, it can be also interesting for us for this biological activity. But when we test the molecule with other type of biological activity assay, we can find other type of property that could be more interesting in fact. Okay. So the message we have to keep in mind at the end of this slide is that these metabolisms are not mainly constitutive, but predominantly inducible. So it means that it is sometimes difficult to discover exactly all the metabolites in a plant. And there is also another idea that we have to understand is that in the plant, the metabolites are not produced in each organs. For example, if you have some metabolites that are produced in leaf after an attack of herbivore, it is not necessary for the plant to produce exactly the same in the roots, because this predator is not a predator of this part of the plant. So what we observe is that, in fact, in each organs, you can find different type of metabolites. This is important, for example, for biotechnological development, because when you want to establish cell lines, it's also important to take for raw material the good organs in order to have the most chance to produce the metabolite you want to produce. Even if the cell have exactly the same genome in each organs, okay, but they have not the same mode of regulation producing, okay, enzymes that are able to produce this type of metabolite. And also, what it is observed, okay, you know that in leaf you have production of this type of one metabolite in particular, but when you enter in the leaf, you can observe that the metabolic pathway leading to the metabolite is in fact divided in different parts that are in different type of cell. So means that you can start your metabolite pathway, for example, in the epidermis, and you can finish it in another type of cells, and very oftenly, as the final products can have a cytotoxic activity, for example, if it is for some defense objective, the cells could be also sensitive, the plant cell could be also sensitive to this molecule, so means that this molecule needs to be accumulated in a particular compartment, in a particular type of cells in order to be not toxic also for the plant itself. So this is why some of plants, they differentiate some type cells such as trichome, sort of small system that you can find at the surface of the leaf, for example, in which they accumulate a large amount of very important specific metabolic. And also inside the tissue, you can have some specific cells named lacticifères, in which you can have accumulation, for example, of alkaloids. Alkaloids are very efficient for the defense system, but they are very cytotoxic. This is why we use them as anti-cancer molecule, for example. So means that in fact, to find the good cells, okay, to initiate the process of bioengineering or cell culture, need to know exactly how the plant is able to produce and where this plant accumulate, produce, and also the metabolic pathway by itself in general. We have seen that they can be soluble, which is more easier to extract and to analyze. Instead, when they are volatile, it's not exactly the same technique used, okay, to capture and also to analyze this type of molecule. And sometimes some are soluble, and just a little modification make them volatiles. So this is important also to have in mind. And when they are volatile, okay, they can be diffused outside the plant. We have seen an example, but then they can also be diffused out the plant, but also they can diffuse in the plant. It's another type of system of working, okay? And the final is that we have just start to identify some of this molecule, because in fact, in one plant, there is several hundred, but now it is estimated between 5,000 to 15,000 molecules by plants that can be produced, not at the same times, okay, but depending of the situation of the plant is. So you see, the potential of one plant is completely amazing. So if you take in mind all the plants that are the diversity of the plant, there is millions of specialized metabolites that can be produced by this kingdom. And not only plants, but fungi have exactly the same property. So we are just in front of a sort of ocean of metabolites that need to be characterized in order to see if one of them, some of them, are for a benefit, okay, of us. So this is why we develop, not we, but the scientific community developed several campaigns of screening in order to identify some type of metabolite that could be interesting for one or two types of biological activity. So we are in front of something that we don't know a lot. specialized metabolites are part of what we call the plant metabolisms. But before arrive to this metabolite biosynthesis, in the cell, there is a normal process, okay, to establish what we call the metabolic pathway. What is the metabolic pathway? Is the successive enzymes that are involved to modify initial precursor to a final product. So it's like in a factory, you have different events, okay, and you need to have access to the first one and the second one and the third one, and you can't go until the end if something is missing in the middle, okay? So means that the metabolic pathway need to be completely coordinated at each step in order to be efficient in the cell. In the cell, it's quite easy because it's a natural thing. When we will see how to do the same in another cells, for example, in yeast, we will see that it is completely different because we need by ourselves to make this coordinate system for expressed all the steps of the metabolic pathway. And before arriving to metabolite, we need enzyme. But the enzyme, you know, are proteins that are encoded by specific genes that are transcribed in mRNA, okay, and before to be translated in proteins. And when you have the proteins need to be completely modified, okay, in order to be active and to do exactly why this protein is produced. So this is quite classical, but when we want to initiate an engineering program, we need to be at each level. Okay, so this is why to have an idea of metabolism is a good idea. This is a start point of the program we want to initiate. But after that, we need to be able to do some molecular biology, genetic activity, biochemistry after, and specifically with proteins, modelization, and also characterization of activity of enzymes, site of activity. It's a very, very transdisciplinary, okay, type of research in order to arrive to produce this metabolite or to understand how the cell is able to produce this metabolite. And what we use now, because we have the chance to have access to the what we call big data, okay, is to develop some genomic research in order to have an idea of what are the gene, the complete number and nature of gene of a genome, because now it's possible to sequence very fastly a genome. But to identify all the genes is another question. It is not possible at this time, but we try to do. But what we use a lot in the type of process we develop, it is the what we call transcriptome and what we call proteome. Transcriptome is less informative as a genome because transcriptome is like a photo at one time, okay, of what the cell expressed from the genome. Means that not all the genes are expressed every time, only a part of them are expressed and are dedicated to produce a corresponding protein. So studying transcriptome is very important because it is also a way to see in what state is the cells. And for example, when a cell is stimulated, okay, for example, by an herbivore, and if the cell needs to produce specific specialized metabolite, at this moment, the gene that encode enzyme of metabolic pathway are stimulated, means that in the transcriptome, we can find this type of mRNA. And by this way, we can also define in which condition we will be in the best condition to identify the enzymes that are responsible of the metabolic pathway. Is it clear? Okay. So means that we will take time together to go in details in these different domains. Okay. So what it is important that if we want to develop some biotechnological systems to produce a specific metabolite, we need to study a lot of things before to have all the tools, molecular tools needed to engineer this type of biotechnological system. You can't see because it's too clear. It doesn't matter. It was just to show the complexity of the metabolism in the plant. Perhaps you can go to see that on the company Roche. They propose a poster in which they have summarized all the metabolic pathways that have been already identified, primary and specialty. And what it is interesting is that you can summarize this by a large system of connection, okay, like in electricity, you can make some network with a lot of connection. For the metabolism, it is exactly the same in the cell. What means? It means that if you modified just one step, it can have lots of consequence in other type of metabolic pathway. This is why, in fact, they are linked, but when you want to develop something and you want to modify just one case, one enzyme or some enzyme, every time you need to be careful because you can have a lot of consequence in other type of metabolic pathway, primary or secondary one, that can affect finally what you want to obtain. So this is important to see. We will see together what are the main metabolic pathway that lead to the main type of specialized metabolite, but they are not alone in a cell, okay? They are linked with a very complex system of pathways for primary or secondary metabolites. So the story starts always with the same systems, the ability of plants to do some, they are unique to do this type of system. It is the photosynthesis. Photosynthesis, it's a way, it's the first chemical modification of one carbon, what we call inorganic carbon, to be integrated in the organic system to become an organic carbon to make some carbohydrates. This is the start point of the story of all the metabolisms of the plants. So it is important to know very well this, and as you can see here, photosynthesis involves not only leaves that are with chlorophyll, with the pigment necessary for photosynthesis process, but also you need the root parts because you need lots of not metabolite but mineral and other compounds that are in the water, kept by the root system to the stem and leaf system. So means that in fact the plant is a formidable system that have take advantage of each organ to produce this phenomenon to transfer carbon from inorganic phase to an organic one. And in the plant cells, if you know that perhaps very well, the photosynthesis starts particularly in chloroplast. In fact, it is not the only organelles needed for making all the different molecules needed to produce glucose at the end. And two other organisms are very important, peroxisome and mitochondrion, that are necessary for producing energy. The energy needed for all the functioning cycle, okay, because each step here are catalyzed by a specific enzymes that need or not, depend of the enzyme, energy to function. So means that in fact, when we take in account production of a specific molecule, primary or secondary, we need to know that the cells need to be able to produce some energy because it costs a lot to cells to produce this type of molecule. So it's time to go to see how it's organized, okay, at the cellular level, a plant cell. So the three main category of the primary metabolite start point that they are completely linked to photosynthesis are obviously carbohydrates with glucose, okay, the main molecules produced by the plants, And after this, glucose can serve to develop the other others of the plant, and also proteins and lipids. Both of these metabolites are well known, we know exactly, but what for us it's interesting is that, in fact, it is this one that is the first point to enter in the system of secondary metabolite. Because, in fact, as you can see here, this molecule is able to produce every amino acid. Okay, you can see tryptophan, phenylalanine, tyrosine, and different other type of amino acid that are the start point of the different metabolic pathway leading to terpenes, alkaloid, and phenolic compounds. So, in fact, it's very important to have an idea how this precursor, because we can name this amino acid as precursors of this specific metabolite biosynthesis pathway, specialized metabolite biosynthetic pathway. Here, for example, is one of the more important, the three tryptophan, phenylalanine, and tyrosine are very important, for example, for alkaloid and for phenolic compounds, and also terpenes in one way, okay, production. And in fact, you see here, I gave you, but it is difficult for you to see, but just also to illustrate it as it is complex. For producing, for example, tryptophan, phenylalanine, or tyrosine, you can see you have two precursors, erythrophosphate and phosphoenol pyruvate. And you see you need 1, 2, 3, 4, 5, 6, 7, 8 steps in order to have the final amino acid. Each step is catalyzed by one particular enzyme. Okay, so you need to have this functional in the cell to have the rest of the metabolite. This is conserved in each cell, means yeast, fungi, animals, plant cells. This is important to know because when we would like to introduce some specific metabolite or specific plant metabolic pathway in a yeast, we need that these cells will be able to produce all these precursors needed to feed finally the metabolic pathway we want to transfer. So it is important to know that. Secondly, what are the three main families that are produced? I have put here, okay, that these primary metabolites are produced normally in a very large quantity. Normally in a cell, there are no limitation with this type of metabolite, which is not the case with the specialized metabolites that are produced in a very smaller quantity, and sometimes they are not produced at one time particularly. So this is one of the most important differences between the two types of metabolites. And after here, you have the three main classes, terpenes, nitrogen compounds, including mainly alkaloids, but in this compound you can find also glucosinolate. I will take time to explain the importance of these molecules, and cyanogenic glucoside too. And the phenolic compounds that are well known also because they produce a lot of type of molecules that are of interest for perfume, for example, for food, okay, for spice, or contains a lot of this type of metabolite, and they make some flavor specifically. And what it is, okay, you can see something, yes. Here it is a link that is now well established between these primary carbon metabolisms and these specialized carbon metabolisms. As you have seen here, photosynthesis is the first point of entrance, and after there is a lot of pathway that leads to the precursor. I show you some just before. And what it is interesting is that these precursors are the starting point of 4 specific routes, means that are specific part of metabolic pathway, named chimic acid pathway, malonic acid pathway, mevalonic acid pathway, and methylerythritol pathway. It is not necessary for you to register, okay, all these names. It is just to show you that in fact, there is only 4 really important pathways that we need to know, because all of these pathways, or alone or by combination, lead to the 4 type of the 3 type of all the metabolic branch leading to specialized metabolite. Here, for example, shikimic acid combined with malonic acid lead to lots of phenolic compounds that are also able to produce only by shikimic acid. And on the other way, all the terpenes, for example, are produced by only 2 metabolic pathways. I will explain more in detail both of them, because at the beginning we thought there was only one mevalonic pathway that is common to all the organisms, microorganisms, and also plants, not plants, oh yes, plants, but also animals, fungi. But in plants in particular, compared to animals and fungi, there is this original pathway that can be also observed in microorganisms. So that leads to the plants the possibility to have 2 origins of terpenes that can also have influence of specialized metabolism in general. Okay. So here are exactly the same representation. So here the core, the heart of metabolism. So with the Calvin-Benson cycle, you see it's the first step of photosynthesis, and also this one. And what it is represented here is that the primary metabolism lead to these 2 types of secondary metabolites, what we call the soluble metabolites and what we call the volatile metabolites, that are every time these volatile compounds come from soluble ones that have been little bit modified. For example, what was presented just before, methyl jasmonate come from jasmonate. Jasmonate is a soluble molecule. Methyl jasmonate become a volatile one. And the only difference is the addition of a methyl group. So by a specific enzyme named methyltransferase to completely change the property of this molecule. Okay, so it is just a general view of what are organized finally this type of metabolic pathways. What it is also important to keep in mind is besides these metabolic classes you see here, lots of other type of molecules are now considered as specialized metabolites. And for example, all the hormones, phytohormones, are now well known as to be specialized metabolites, even if they are more ubiquitous, in different type of all plants are able to produce acid abscisic acid, jasmonates, and also auxin and so on. But they are derived from the pathway that are included in specialized metabolic pathway. And one point on which it will be difficult for us to enter in this lecture because it's really too long to introduce, it is also the importance of receptors. Because you will see that this metabolite, to be transformed from a precursor to the final one, they need to change of compartment in the cell, and sometimes to the cell to another type of cell. And the route to go from one point to another one needs to go across a several number of membranes that are very different. So they need to be transported by this receptor and also this transporter in order to be led to the good point. So this can, we need to adapt all this in a biotechnological process if we want to develop. And the final one, sorry, the light is too high. Okay, this is a very interesting point because now there is a lot of study that emerge that try to know from where and when this metabolic pathway leading to this very large amount of specialized metabolite have been implanted in plant cells. And when we are looking at some specific metabolite in what we call the first plants, such as algae, for example, mosses or pteridophytes, we see that not all these plants are able to produce a large amount of metabolites. Why? Because the metabolic pathway are more simple in these primary plants than in the angiosperm or gymnosperm that are considered as the last one, that are the more evoluted plants. And why? Why? Because in fact, what it is interesting is that, as I said to you, primary metabolites lead to a specific, in particular, amino acid that are taken charge by specific enzyme to be transformed in specialized metabolites. But what happens during the evolution is that one enzyme normally is known to recognize one substrate, to catalyze one reaction in order to give one product. This is what you have studied in class. But in the reality, it is not the case. One enzyme can recognize different type of substrates, and then they can also catalyze different type of reaction. So this is very complex because when, for example, a chemist says you have to add here a methyl group, but a methyltransferase, okay, is able to do that, but other type of enzyme are also able to do exactly the same activity. This is the first point. It is what we call promiscuity of the enzyme, the ability to have several substrates. And there is also another point, because during the evolution, what happens in plants? They have duplicated a large part of their genomes. Means that they have not only one copy of gene that lead to one enzyme, but they have a large amount of a number of copy of a gene that encode for a number of the equivalent number of what we call isoenzyme, because it is enzyme that have quite the same sequence, that they have the same activity, but they have the possibility to recognize a most important number of substrates. So by this way, you can have a large diversity of modification that lead to a bigger diversity of intermediates or final products. So this is important because in all the angiosperm and in all, mainly in angiosperm, for one bioconversion, biochemical modification, you have not only one possibility of enzyme, but you can have a lots of plurality of enzyme. Sometimes, but as a contrary, sometimes they are very selective. So you never know what would be exactly the good enzyme that are able to produce one particular step of your metabolic pathway. Okay? Is it clear for all of you? Perfect. So it means that to screen the metabolites, it's good, but to screen also the enzyme that lead to this metabolite, it is another work. A very, very important one, because if you don't have these tools, you can't develop any program of bioengineering, even in a cell, plant cell, or in yeast cell. You need to have this knowledge. Okay, it's a prerequisite idea to enter in a program of bioengineering. And as it is difficult to find the good enzyme, it is also difficult to find the good substrate, to have an identification of each step of a metabolic pathway. Just, okay, a metabolic pathway can have 2, 3, 10, 40, 50 steps. So from the precursor to the last product, you may have a lots of works to do in order to identify and to decipher completely this metabolic pathway. It is one of the expertise of my lab. Okay, I will show you at one time what we are doing. So here are all the plant, the organization of the different metabolites. I like this picture because you can't see, because it's not so nice with this projector, but you will see that the scaffold of each molecule are completely different. So means that in fact, the diversity, the chemical diversity of this molecule are very important. Even if you are, for example, in one group, such as alkaloid, not all the alkaloids have the same structure. So means that not all of them have the same biological activity. It is the same if you look at some glucosinolate or terpenoids, terpenes, and phenolic compounds also. So what we know is that, in fact, the estimation is about 1 million compounds, but it is, I think, less than the reality. Perhaps more millions of compounds are available in the nature. And what we have already characterized, it's about 50,000 structures. So it's quite very small part only for the metabolite, and also only in the diversity or biodiversity of plants, because only 20 to 30% of the plant species have been started to be studied according to their ability to produce specific metabolites. So there is a large work to continue to do. But now we are in a period in which new techniques and possibility to access to omic data have accelerated the process of identification of enzyme and metabolic pathway at a level you can't imagine. For example, it was necessary about sometimes 10 years to identify one enzyme. And now, in 1 year, you can, if you have a little bit some chance, okay, you can access to about 10 different enzymes. Okay, so you see, it's not the same temporality for having this type of information. Wide variety of structure, it is what I said. Then, and the last point is important. In fact, all the plants, are able to produce terpenes, alkaloids, and phenolic compounds, but not the same. But what it is interesting is that sometimes some specific, for example, phenolic compounds, depends on specific families. So means that if you are looking for specific enzymes that are involved in their biosynthesis, you can obviously look at the plants that are producing the metabolic you want, but also you can see what happens in the other member of the same family, because they may have some enzymes equivalent, more efficient, or with affinity, different affinity with different substrate. So it means that sometimes it's interesting to look at one plant, but also all the members of the same family in order to have more information of how organize this metabolic pathway. And so it is what we use currently. So this was the general consideration of what is a metabolic pathway and why it is so difficult, in fact, to identify them correctly because of this lots of varieties of enzyme and the metabolite.